Good evening. How are we doing?
I'm doing fine. Don't know about you, sir.
I'm fine as well. Thank you. Okay, so quickly, let's dive into our tutorial for today. And we are starting looking at genetics. And the first topic that we're going to look at is what is known as the chromosomal theory of inheritance. The chromosomal theory of inheritance. And in this lesson, we're going to look at the theory itself. And then we're also going to look at the discovery and the structure of the chromosome. The discovery and the structure of the chromosome. And so we are going to touch a bit about a bit of history so that we can actually have some glimpses on how the discovery of chromosomes and inheritance in general came to be. So we're going to consider some very good names in biology or in science that are closely linked to genetics and the various theories and discoveries and laws. So on the discovery of chromosomes, I'm going to consider certain names such as Gregor Mendel, and the others such as W. Wardayer, which are famous names. So we find Mendel proposing laws of inheritance that are independent of gametes in the 1860s. And we're actually going to look at one of the lessons that we're going to look at in genetics will be Mendelian genetics, classical Mendelian genetics. So he's a very good and prominent figure in the field of inheritance, in the field of genetics. We also find the discovery of the process of cell division in somatic cells, otherwise known as mitosis, being discovered also in the 1870s. Also find the reduction cell division in the formation of gametes, otherwise known as meiosis, also being discovered in the late 19th century as well. And these, we are going to look at these, the mitosis and meiosis, when we look at cell division, which will be our next lesson. And then in the 1900s, we find Mendel's classic papers being discovered by Correns, de Variès, and Cercenage independently. So they found Mendel's papers were rediscovered or discovered by these gentlemen. So these are some of the prominent men in this field. Now we see that these are speculations about the chromosomes being key to understanding heredity. Remember Gregor Mendel discovery. And so we find that scientists started reviewing or examining Mendel's publications to be able to reevaluate his model in terms of how chromosomes behave during cell division. So they started those speculations, or rather examinations of Mendel's publications. We have the man Theodore Boveri. This was a German scientist who actually discovered that the number of chromosomes was reduced to half in the gametes. So in the gametes, they discovered that they were actually reduced in half during the process of meiosis. So Theodore Boveri discovered that the number of chromosomes was reduced in the gametes during the process of cell division, otherwise known as meiosis. That's Theodore Boveri. So he was an embryologist. So he observed that proper embryonic development of the CU cairns does not occur unless chromosomes are actually present. And then we also have the man Walter Sutton. We have the man Walter Sutton, who was an American scientist who observed the behavior of chromosomes during meiosis. And he noted that the behavior of these chromosomes during the process of meiosis actually agreed with Gregor Mendel's second law of inheritance. He agreed with Gregor Mendel's second law of heredity. So that's Walter Sutton. So he observed that the separation of chromosomes into daughter cells during meiosis. Now, these observations led to the development of what is known as the chromosomal theory of inheritance. And this theory identifies chromosomes as being the genetic material responsible for Mendelian inheritance. So according to this theory, we see that chromosomes are actually the carriers of the Mendelian factors, and meiosis is the basis of separating the factors into okay. Sorry about that. Network issues. So we're saying that the chromosomes are actually the carriers of Mendelian factors, and meiosis being the basis of separating factors into the gametes. So actually before DNA and chromosomes were actually known, we find that actually the field of genetics was already discovered. And then we find in the 1900s, or in the 19th century rather, in the late 19th century, we see, or we finally saw the chromosomes. So the chromosomal theory of inheritance was actually consistent with Mendel's laws and was supported by the following observations that were made. So you find that during meiosis, we have what are known as the homologous chromosomes. The homologous chromosome pairs migrate as discrete structures that are independent of other chromosome pairs. So these observations were observed during the process of meiosis. And then there was also an observation of random assortment, or the sorting of chromosomes from each homologous pair into its pre-gametes appears to be random. So we have random assortment being observed. And then the other observation that was made was that each parent synthesizes gametes which only contain half of their chromosomal complement. So they're just going to contain half of their chromosomal complement. The other observation was that even though the male and the female gametes, otherwise known as the sperm and the egg respectively, differ in the size and shape or morphology, they actually have the same number of chromosomes, which led to a suggestion of equal genetic contribution from each parent. The other observation was that actually the gametic chromosomes combine during the process of fertilization, which leads to a production of offspring which contain the same number of chromosomes as their parents. So these observations were made. We may be familiar with the Drosophila melanogaster. It's one of the lab sessions that are involved in the course of biology at first year, where we look at variation, genetic variations, and the specimen that is being used is the fruit fly Drosophila melanogaster. So we may be familiar with this. We have the man, Thomas Morgan. Now, Thomas Morgan was actually an American scientist. Thomas Morgan was an American scientist, and he showed clearly that the white color of the eyes in Drosophila was due to a mutation. And this mutation was located on the X chromosome of the Drosophila melanogaster. So we see that there are actually this comparing correlation that existed between the behavior of chromosomes during meiosis and Mendel's abstract laws. We see that the chromosomal theory of inheritance was proposed long before there was any direct evidence that traits were actually carried on chromosomes. So after years of carrying out crosses with the fruit fly, we find that Morgan provided experimental evidence which led to the support for the chromosomal theory of inheritance. And then we are very familiar with DNA. We looked at DNA when we looked at nucleic acids, and we find that DNA now being discovered. And remember the Watson and Crick experiments coming up with what is known as the Watson and Crick model, DNA being a double helix, and also confirming, actually confirming Taggart rules as well, that the proportion of purines to pyrimidine will always be equal. And that means that guanine and cytosine will always be bonded via 3 bonds, and adenine and thymine will always be bonded via 2 bonds to each other. And we also find the contribution of Rosalind Franklin and Morrison Wilkins as well. So also find the discovery of DNA. Now, what is the structure of the chromosome? What is the structure of the chromosome? So remember, we looked at the eukaryotic cell, for example, and also the prokaryotic cell, and we looked at the organelles. We looked at the organelles, and among the organelles that we have is the nucleus, and we said that the nucleus actually contains the genetic material. So when we look at the prokaryotes, we're going to see that actually for the prokaryotes, the genes are located in the cytoplasm. The genes will either be on the chromosomal DNA or the genomic DNA, or on an extra chromosomal DNA, which is known as a plasmid, if we can recall on our lesson on prokaryotic cells. When we look at eukaryotic cells, we see that actually the genes could either be located in the nucleus on the chromosomal DNA, or on the cytoplasm, on the mitochondria. Remember, mitochondria has DNA, and chloroplast also has DNA as well. So in prokaryotes, the chromosomes are actually made up of circular double-stranded DNA. So no wonder the other point is that they're actually described as being naked because they do not contain any proteins. However, when we look at the eukaryotic cells, we find that there are actually several chromosomes, and these several chromosomes contain DNA, which is tightly bound to proteins. So eukaryotic chromosomes actually have equal amounts of DNA and proteins as well. So the chromosomes are actually the carriers of the genes, and they represent the material basis for inheritance that we've been looking at. So the chromosomes carry DNA, in other words. And we know that DNA is a genetic storage material, and also is able to transmit that genetic material or information from one generation to another, So there are actually many acidic proteins, or rather there are many kind of proteins that are associated with DNA. However, of importance, of course, 5 basic proteins. So these are what are known as histones. So going back to our lesson on nucleic acids, we did look at the histones, or the DNA being associated with the histones. There are 5 types of histone proteins, which is, for example, H1, which is packaging into which is essential for packaging into solenoid, and we have H2A, H2B, H3, and H4, which to which DNA is going to wrap around that. And then we find that each chromosome has main fundamental units, and these fundamental units are what we call the nucleosome. These are the nucleosome, and it contains 8 units, or the 8 histones. So the nucleosome appears like a bead structure when we look at it under the microscope, like a bead. It appears like bead structure and consists of 8 units of 2, each having H2A, H2B, H3, and H4. And this is going to be wrapped around, and DNA is going to wrap around, and making what is known as the 1/3 tens. And the H1 is, like I mentioned, for packaging into solenoid. It's a spacer. It acts as a spacer. So that's the chromosome there. That's a chromosome. We see what is known as the centromere, which is a structure in a chromosome, which is going to hold together the 2 chromatids. So we have the chromatids, the sister chromatids, which in short, these are daughter strands of a replicated chromosome. So when a chromosome has duplicated or replicated, it's going to form the sister chromatid. So a sister chromatid refers to an identical copy formed by DNA replication of a chromosome. An identical copy formed by the DNA replication of a chromosome. And both of these copies are linked together by a common centromere. So in short, we are saying, in other words, we may call a sister chromatid being 1/2 of a duplicated chromosome, being a 1/2 of a duplicated chromosome. So a full set of sister chromatid is actually created during the S phase or the synthesis phase of interphase, during which all the chromosomes are replicated, all the chromosomes in a cell are actually replicated. So a pair of sister chromatids is actually called a dyad. A pair of sister chromatids is called a dyad. So there's a centromere, a sister chromatid. So the centromere holds the two chromatids together. It's a specialized DNA sequence of chromosome, which links a pair of sister chromatids. It provides the foundation of the assembly of what is known as a kinetocophore, or rather the kinetoco. It lays the foundation for the assembly of the kinetoco, which is here. It lays for the foundation of the assembly of this structure known as the kinetoco, which is a complex, a protein complex, which is essential for proper chromosomal segregation during the process of mitosis. So as we can see, the centromere is a constricted region in this chromosome. It's a constricted region. And in addition, it separates the chromosome into a short arm. You can see there's a short arm and a long arm. So the short arm is usually referred to as the P arm, or the long arm is referred to as the Q arm. So it separates into the short arm and the long arm. So what happens during the process of cell division, as we are going to see, is that the chromosome will actually first replicate such that each daughter cell will receive a complement set of the chromosomes. And then following DNA replication, hence the chromosome will consist of these two identical structures, which we have already described as being known as the chromatids. And these are formed at the centromere. So this is a loci. The centromere is a loci, and it's going to mediate the equal distribution of the duplicated genome between these daughter cells. So this is a centromere. And then we also have, we can also see the telomere. Of course, we are going to explain about the telomere later. I'm going to explain about the telomere later. And then we have what is known as the chromatin. We have what is known as chromatin, which is which makes up the chromosome. So chromatin makes up the chromosome. So the chromosome is made up of chromatin. And then chromatin is made up of DNA, RNA, and proteins as well. So what happens is that at interphase, remember at interphase, the chromosomes are actually visible. They're going to be visible as thin chromatin fibers that are present in the nucleoplasm. And then during the same process of cell division, the chromatin fibers condense, and the chromosomes are actually visible with distinct features as well. So you have chromatin there. Okay, so when we look at the chromosomes in humans, we see that each cell actually contains two versions of every single chromosome, as we have already explained. So we have, for example, maternal and paternal. Maternal, mother, paternal, father. These have the same genes, but they are actually not identical. So we have cells that are known as the diploid, di for 2, so it's 2n. And we have cells which are known as the haploid cells, So 2 copies of each of the 23 chromosomes will make a total of 46 chromosomes in the human cells, the diploid cells. So 2 copies. Remember, we talked about homologous chromosomes as well. And then we have genes and alleles. Genes and alleles. So remember, a gene is a segment of DNA which codes for a complete polypeptide. So genes could either be structural genes, and this means that these are going to code for for functional proteins. For example, enzymes, hormones, membrane proteins, the antibodies, the storage proteins. And then we also have regulatory genes, and these simply serve to control and regulate the activities of the other genes. So we have genes and alleles. So when we look at an allele, okay, here it's showing a homologous pair of chromosomes, homologous chromosomes. So when we talk about homologous chromosomes, this is simply a pair of chromosomes that contain the same gene sequence, each derived from one parent, making what is known as a homologous chromosome. So we have, in our example, we have allele 1 and allele 2, and we have sister chromatids there, which precisely have the same genes and precisely the same sequence of nucleotides. The sequence of nucleotides, however, will not be identical. Remember, they will not be identical. You may have the same genes, but the sequence of nucleotides will not be identical. When we look at the alleles, or otherwise known as the allelophones, or alleles in short, which is simply alternate forms of a gene. These are going to occupy an identical loci on homologous chromosome. They're going to occupy an identical loci on the homologous chromosome. So these actually control contrasting characters of the same trait. They're going to control contrasting a character of the same trait. So usually they exist in 2 different forms. So these 2 forms of the same genes are what are known as the alleles, or the allelomorphs, of that gene. So for example, we can give a gene which codes for height. So a person will either be short or tall. So you have an allele for tall, you have an allele for short. We can also give an example, an example of blood groups. You can have an allele of blood group A, an allele of blood group B, and an allele of blood group O. So there are 2 forms of the same gene. They control the contrasting character of the same trait. For example, height, for example, blood group. That's an allele. Like I mentioned, the gene is going to code for a protein, as we have mentioned. So homologous chromosomes, or the homologs, have the same genes, but they are not actually identical. And then we have the sister chromatid that we've already actually explained. There. And then we have non-coding DNA. Remember, the gene is a segment of DNA which is going to code for a complete polypeptide. So non-coding DNA. The non-coding DNA will not code for amino acids. And usually this non-coding DNA will lie between genes. So for example, here it's showing gene, and that gene. So the non-coding DNA will lie between genes on the chromosome. So this non-coding DNA may function as to transcribe RNA, except for the messenger RNA. They can serve as origins for the process of replication, and important as well is that actually involved in gene expression regulation. And also it comprises of centromeres and telomeres as well. So that's non-coding DNA. Now looking at the telomeres, the telomere is actually simply the end of a chromosome. They are ends of a chromosome. And they are actually made up of repetitive sequences of non-coding DNA. They're actually made of repetitive non-coding DNA. So we're going to look at the function of the telomere, which includes protecting the chromosome from damage. So I find that each time the cell divides, each time there's cell division, the telomere is going to shorten. So I find that as the process of cell division continues, the telomeres also continue to shorten. So there's progressive shortening of the telomeres. So as one ages, actually, the telomere length shortens as well. So telomere length shortens with age. So eventually you see that the telomere becomes so short that the cell can actually no longer divide. The telomeres become so short such that the cell can no longer divide. So as there is progressive shortening of the telomeres, it leads to the process of replicative senescence. It leads to the process of replicative senescence, which simply entails an irreversible arrest of the cell proliferation and outer cell function. Due to senescence, there will be an irreversible arrest of cell proliferation. So the cell will no longer be proliferating. And also there will be an altered cell function due to the senescence. The other things that progressive shortening also leads to programmed cell death, otherwise known as apoptosis. The other thing is that progressive shortening will actually lead to oncogenic transformation of somatic cells, which is going to lead to affecting the health and the lifespan of an individual. So find that cancerous cells can come in. So oncogenic transformation means a cell is being transformed from its normal function into a cancerous cell. So telomeres, being specific DNA protein structures found at both ends of each chromosome, protect the genome from nucleotic degradation, as I mentioned earlier, to also protect the genome from unnecessary recombination and repair, and also interchromosomal fusion. So the telomeres are actually important. As I mentioned, they protect the genome from nucleolytic degradation. It protects the genome from unnecessary recombination and repair, and also interchromosomal fusion. So they play actually an important role in